Okay, so nigrostriatal, this is the direct pathway. Okay, this is normal. No changes, no pathology here. This is just normal physiology, okay? So you're thinking about, in the absence of thinking about movement, this neuron is always on. It's always depolarizing to cause the release of GABA in this space, okay? That means that you're not going to have any movement, right? That's without moving. Now, you think about moving, coming from here, your motor planning area. That thought about moving activates or depolarizes this neuronal cell body that is sending out axons to the striatum, both the caudate and the putamen. Okay? Those axons are releasing glutamate at this space, okay, causing the firing or depolarization of the next neuron in sequence. Normal physiology, you also have dopamine releasing neurons coming from the substantia nigra that are going to contact both the caudate and the putamen, both the striatum, okay, the striatum there. That's also going to cause depolarization. So the combination of glutamate and dopamine combined with dopamine binding to D1 receptors is going to cause a very strong depolarization of this neuron, causing the release of lots of GABA in this space. If there's a lot of GABA released in this space, then this neuron that's almost always, it is always on in the absence of movement, is going to be inhibited because it's GABA. GABA inhibits. It's an inhibitory neuron. If this neuron is inhibited or hyperpolarized, you're going to have a decrease in the concentration of GABA at this synaptic space. Decreasing GABA here is going to result in the release from inhibition of this thalamic neuron that's going to be now allowed to fire, saying it's okay to move. Yes, John.
Okay, so the decrease of GABA, because GABA is inhibitory, causes the firing to move. Basically, like the communication, it's okay to move.
Yes.
Okay.
Yes. This thalamic neuron is released from inhibition. It is normally inhibited by this neuron. But both the combination of dopamine and the glutamate cause this neuron, which is ordinarily on, to hyperpolarize, decreasing the concentration of GABA in the thalamus, allowing this glutamatergic excitatory neuron to fire, saying it's okay to move. This is important for initiation of movement. Does everybody remember what patient population? Oh, sorry. Go ahead, Ben. Did you have a question?
No, go ahead, please.
What patient population did I say had trouble with initiation of movement? Parkinson's. They have a problem in this pathway. We're going to look at that in a minute. But Ben, question?
Yeah. So once you're already moving, is this loop continually active while you're thinking about continuing to move, or is it just like once you thought about moving and you get that movement from the neuron in the thalamus, that's the end of the...
These pathways, so I'm oversimplifying it for the purpose of explanation. These pathways are both, we haven't gotten to the other one. There's a direct pathway and an indirect pathway. They're both always active normally, and it's going to be the amount of firing between each of them that determines what looks like normal movement. Okay. The first patient population we're going to look at is Parkinson's. Parkinson's patients have a problem with this pathway. They also have a problem with the other pathway too, but we're not going to touch that. Direct pathway Parkinson's, that's what you said. Indirect pathway, we're going to look at Huntington's, and then the changes that cause Huntington's. So that they're always going, okay? And it's just the concentrations of neurotransmitters at each of the spaces is going to make a difference in the movement that's produced. Does that help? And I think when we look at the patient populations, you'll see it. Okay, Gabby?
That's fine.
Well, yeah, right here. No, the amount of, so remember, if this neuron in the GPI is always firing, so that means there's GABA here that's inhibiting the thalamic neuron from firing. But if I inhibit this neuron from firing, the concentration of GABA in this space is going to go way down, allowing this thalamic neuron to fire. There's a huge amount of GABA. The GABA is coming from the got both glutamate and dopamine binding to D1 receptors, causing depolarization of those two neurons. Okay, those two neurons coming together are both acting on the GPI. This is causing these two neurons are releasing a ton of GABA right here because of the combination of dopamine and glutamine in that. What has to happen to have not a lot of GABA here? Ordinarily, if there's no thinking about movement, then you're not going to have GABA in that space. Because remember, that neuron is always on. It's always firing because it wants to inhibit movement if you don't want to. Okay. You're welcome. Okay, other questions, Bill?
But then in that case, the GABA would be high where it's low on this picture.
Correct.
So that we don't win.
Exactly. Exactly. Because that was always on. And that's a little confusing, but once you get through the whole thing. Okay, everybody, any more questions about the direct pathway? Okay, like I said, this is going to be important for initiation of movement, and you can see why, right? Now, if we go over to what the next one is going to be Parkinson's disease. So, Parkinson's disease is a very specific problem. Which one do I want? Okay. So, for Parkinson's disease, let's see if I can. This is going to be a problem with the direct pathway. Okay, now you're going to see why. So we can go through and quickly label all this stuff. So this is going to be my caudate, right? The caudate nucleus. And my putamen, collectively known as the striatum. Okay, here's my GPE and my GPI. Here's my thalamus. Here's my subthalamic nucleus. It doesn't play a role in the direct, but it does in the indirect. And then we have my substantia nigra. Okay? So, nothing more than just labeling. And also I had up here, I don't remember what color it was, but just put it in black. This is going to be my motor planning area. So this is a combination of my SMA and my PMC. Okay? My motor planning. Okay. Now, in contrast to normal physiology, which we just looked at, this is going to be Parkinson's disease. Okay. Parkinson's disease is, what happens is this substantia nigra starts to deteriorate. Okay, so if we want to make it simple, just say there's nothing coming out of there. So what came out of the substantia nigra before that was part of normal physiology? Dopamine. Great. Okay, so Parkinson's disease is a decrease in the normal amount of dopamine. Got it? All right. So the pathway is the same. We're going to have, we'll just start from the beginning since we've seen the whole thing. So up here you have a glutamatergic neuron in your motor planning area. Sends out an axon to the striatum, so both to the caudate and to the putamen. Okay? And that's connected up with a GABAergic neuron located in the caudate and the putamen. It's going to be hooked up. The axons are going to come together and synapse with this GABAergic neuron in the GPI that we said was always on. And then you have the neuron in the GABAergic neuron in the GPI here hooked up with the glutamatergic neuron in the thalamus. What we don't have anymore, we're lacking, so we're lacking the input of the dopamine coming into the system. So there's going to be no dopamine coming into the system at all. So that means, thinking about moving, there's going to be a release, there's going to be an increase of glutamate in these spaces. Okay? And these neurons in the caudate and putamen still have the D1 receptors, but there's no dopamine coming into the system to stimulate them. Right? Does everybody see that? So ordinarily, I'd have the combination of dopamine and glutamate stimulating here, causing depolarization of these two neurons. So compared to normal, how much GABA is going to be released here? Is there going to be more GABA released or less? Less. Perfect. So where we had a bunch of GABA before, now we have just a little bit of GABA. So if I have a little bit of GABA here, if there's less GABA here, the relative concentration of GABA that's coming out of here is going to be more. And if I have more GABA here, can you see that I'm going to have less firing here and I'm going to have a decrease in movement initiation. The reason is because there's no dopamine in the system. Usually, normal physiology is dopamine and glutamate causing major depolarization of those striatal neurons. Okay, but you have less GABA being released here compared to normal, which means there's more GABA here, which is helping to inhibit that movement. So you're having decreased initiation of movement. It's very specific for Parkinson's. Questions? Yes, Ben?
So it's not that there's no GABA being released now that there's no dopamine, it's just less than what we had before?
Absolutely.
So why does that cause there to be increased GABA in the thalamus?
Because having less GABA at this position doesn't completely hyperpolarize this neuron.
Okay, since it isn't completely hyperpolarized, you still have more GABA.
Yeah, it's the concentration of neurotransmitter at that space that makes the difference. Okay, normal physiology, you get a double whammy. I like to think about D1 receptors and glutamate. We're going to fire big time. We'll talk about this a little bit more, and I'm sure Dr. Blair will talk about how the medication that's appropriate for patients with Parkinson's. What do you think they would want to do with patients with Parkinson's as far as medication is concerned? Dopamine, yeah, some sort of dopamine, and we'll talk about that as we go forward. Yes, please.
So as far as like the patient presenting with Parkinson's, how is it that they like get over the hump and initiate the movement? Is it just like the GABA essentially builds up?
Good question. One of the things that you'll kind of putting it all together in a neurorehab perspective as well, which you'll get there, but the best thing I can say to you is it's the thinking about movement, that internalization of thought, that is the problem. Okay? Because if you ask a Parkinson's patient to have an external focus of control, they can move. So it's when they're constantly struggling and thinking, why can't I do this? Why can't I do this? But I know if I have a Parkinson's patient and I put like a cone on the floor or a piece of tape, and I'm asking that person to move or step, just let's walk. I know if I put a piece of tape down and I say step on that tape, they can do it. That's what starts it. It's that change in focus. It's that thinking that is the problem. Not a problem, but that's the best example I can give you. Does that make sense? Yeah. It's really hard for them to get started. And we'll see videos as well. Okay, other questions? Yes.
So the whole deciding to initiate the movement. Like that's the thought of initiating movement is still happening. It's just like because there's no dopamine and then there's more GABA. It's just harder for them to achieve movement.
Correct. Yes. It's the change in concentration of neurotransmitters at each space from normal. Okay? Just think about the change in concentrations of the neurotransmitters. Other questions? Does everybody see? Compared to normal, does looking at Parkinson's help make sense of the other as well? I thought it might a little bit. But this is Parkinson's disease. Okay? Problem with dopamine. What happens is the substantia nigra starts to degrade, and there's lack of dopamine in the system. It is a progressive disease. That means that patients are going to get worse over time. Patient population we see all the time. Lots of tricks to working with them. Any questions about? Yes, Sydney.
Sorry. So
That's
is
fine.
it that there's no dopamine, or is it that there's less?
Oh, so initially there's less. So we'll look at videos, but initially they start out with one-sided with a problem, like one side won't move as well as the other side, and then over time it becomes less and less dopamine. Okay, there's only so much. Well, that's more than we'll talk more about that next year. Okay, other questions? Okay, that's Parkinson's disease. This is the direct pathway. Okay, Parkinson's is a direct pathway problem. See if I can find the next one. The next one is we're going to talk about the indirect pathway. Okay. Let's see if we can get there.
Dr. Basal,
Yep.
sorry to bother.
No, don't bother. Ask me questions, please.
About the Parkinson's,
Mhm.
why again is there an increase in glutamate at the synapsing with the GABA?
Why is there an increase in glutamate synapsing? Hold on a minute. Let me pull up the Parkinson's. Now, why is there an increase in glutamate here, or
Yeah, like at both
Here?
of those, you said there's an increase in glutamate at both of the...
Because you're thinking about moving, okay, and thinking about movement depolarizes this glutamatergic neuron, which releases glutamate here.
So the thinking about causes the depolarization, which causes more glutamate to be...
Yeah, not more. It causes glutamate. It causes this neuron to depolarize, causing release of glutamate at that space. Okay? But realize that ordinarily you'd have glutamate and dopamine. You only have half as much of a let's get started signal as you're used to in the Parkinson's case. Does that make sense?
Yes.
All right. Anybody else? Moving on. Okay. Let's look at the indirect pathway. The indirect pathway is a little bit different. It's called the indirect pathway because you're not going from directly from the GPI to the thalamus anymore. You're going to make a detour down through the subthalamic nucleus and then back up to the GPI. It's the only difference. So it's indirect in its navigation to the thalamus. Same players, okay? It's going to be my caudate nucleus. Okay, here's going to be my putamen. Okay, here's my GPE, GPI. There's my thalamus. Subthalamic nucleus, like I said, subthalamic nucleus will come into play here. And again, we have our substantia nigra down here. Dopamine producer. Okay, not doing anything but just labeling. And then, as before, you have your SMA and your PMC at your motor planning area. Let's see that, okay? Okay, let's go through and look at the indirect pathway. So, for the indirect pathway, you're going to have The indirect pathway is going to be important for two things. You're going to be important for inhibition of unwanted movement and also the amplitude of movement. Okay? Which is going to happen is going to depend on the relative concentration of neurotransmitters in the beginning. So what happens is you have a thought about movement, so the same glutamatergic neuron sitting here in the motor planning area, and it sends out axons to both the caudate and the putamen. Okay. It synapses with glutamatergic neurons in both the caudate, GABAergic neurons in the caudate and the putamen. Okay. Instead of going directly to the GPI, it gets there indirectly through way of the GPE. So there's going to be another GABAergic neuron present in the GPE. So you have axons coming here from the striatum, collectively, both the caudate and the putamen, okay, making connections with that GPE. Okay, now the GPE, GABAergic neuron, sends out an axon here to the subthalamic nucleus, where it is hooked up with the glutamatergic neuron, glutamate releasing neuron, and that glutamate releasing neuron synapses with the GABAergic neuron and the GPI. So that's the indirect pathway. Took an indirect path to get there. And this GABAergic neuron is hooked up to the thalamus, and then this is the feedback loop communicating with the SMA and PFC. Okay, with the indirect pathway, let's look first at the indirect pathway in the absence of any input from dopamine. So the other thing that I didn't draw, oh, there she goes. Okay, the one thing that I didn't draw in here yet is there are axons coming out of the substantia nigra that are dopamine releasing, and they're going to make contact with these neuronal cell bodies in the striatum again. Okay. But in contrast to the direct pathway, the indirect pathway is going to bind here to D2 receptors. Okay. Like I said, what happens in this pathway is going to be dependent on the amount of glutamate compared to the amount of dopamine released here. Okay? Because remember, are D2 receptors inhibitory or stimulatory? Inhibitory. So we have two things happening. Coming from the motor planning area, you have glutamate, which is going to be stimulatory, right? Going to cause depolarization. But then you also have dopamine coming into the system binding D2 receptors, and that's going to be inhibitory, right? So what happens in this pathway is going to depend on the amount of dopamine in this position. Does that make sense to everybody? Okay, so let's just say, starting out, we're going to look at the pathway when we're going to have a lot of glutamate being released here. We'll do that first. Okay, so we're having a lot of glutamate being released here. And I'll put that in red. And we're going to have, let's just even say, for the sake of argument, a really low level of dopamine. low level of dopamine there. So everybody got what I'm trying to set up here. So I'm going to have, there's a competition here. It's the amount of glutamate versus dopamine at this position. Okay, and all we're going to go through, right now we're going to look at a situation where you have a lot of glutamate being released and not very much dopamine. Let's look what happens with the indirect pathway. So if I have a lot of glutamate being released here and it's hooked up with GABAergic neurons, what's going to happen at this position? Is GABA going to be released or not released? Released, right. So I'm going to have an increase in GABA at this position. Oh. I'm going to have an increase. And this is going to be a situation where there's a little bit of glutamate and not very much dopamine. Okay, so I'm going to have GABA released here. If GABA is released here, is that going to cause this neuron to fire or not fire? Not fire. So I'm going to have less GABA here. Okay. If I have less GABA here, and then this is a glutamatergic neuron, is this going to depolarize or hyperpolarize? Depolarize is going to cause an increase in glutamate here. Okay, so I have an increase in glutamate here. Okay, if I have an increase in glutamate, am I going to cause GABA to be released in this position or not? Yes, so I have an increase in GABA there. So I have an increase in GABA. Okay, if I have an increase in GABA here, is this going to cause this neuron to polarize, to fire or not? Am I going to? No. So I have a decrease in firing, so we're going to have a decrease in movement with the indirect pathway. This is the indirect pathway that's biased toward glutamate. There's more glutamate in the system. Okay, so to kind of make that a little bit clearer. Okay, questions about this one? Hopefully this one's a little bit simpler after going through the direct. Okay, so what I'm going to do is I'm going to put a square around the things that we just did. All these squared things are going to indicate the glutamate versus dopamine. Okay, so here if it's surrounded by a square, that's an increase in glutamate and a decrease, less dopamine. So in other words, the system's going to be very active because there's not a lot of dopamine to stop it. Okay, so when I do that, I have an increase in GABA here. Okay, I have a decrease in GABA here. I'm going to have an increase in glutamate here, increase in GABA, and also a decrease in movement. Okay, and again, this is going to be biased toward glutamate predominant. Questions, Ben?
So you said that there's not a lot of dopamine to stop it because we have D2 receptors, which means that the dopamine is going to inhibit.
Dopamine's going to, correct. Dopamine in this system is going to inhibit it. What I want you to see is this is going to be a situation kind of like it's going to depend on what your intent is, but there's not going to be a lot of dopamine in the system. And an example of when you want that to happen is when you're doing some sort of fine motor activity. Okay, so you want to inhibit unwanted movement. Okay, so neurotransmitter concentrations are going to be adjusted depending on what your intent is for the movement.
So
Okay?
like, say like a surgeon or artist or someone would be using more like indirect pathways.
Yeah, in the indirect pathway that's going to function with that in mind would be more glutamate in the system and less dopamine in the system. Okay, that's regulated by your CNS. But there's two things that need to happen here. There's going to be amplitude, which is large versus small, versus stopping unwanted movement. Okay? So this is an example when you have glutamate predominating in the system over dopamine, then you're going to have stoppage of unwanted movement. Okay, other questions? Okay, and it's the glutamate, the amount of glutamate in the system that really makes that happen. Can everybody see that? Right? So if I had a lot more, we're going to draw here in a minute what the dopamine would look like. Okay? If you have an excess of dopamine in the system, what happens? Okay? So it's really the relative concentration of glutamate to dopamine here that's going to make the difference. Now, if, let's say, I have more dopamine entering into the system. Okay? Can you see? If I have more dopamine entering into the system, I'm going to hyperpolarize this neuron. Does that make sense to everybody? Okay. If I hyperpolarize this neuron, am I going to have, so let's go ahead and write that down. So this is going to be, I'll see if I can circle it to make it more sense. So let's look at a situation when we have a decrease in glutamate and an increase in dopamine. Okay, and for those, I'll go ahead and circle it in purple. See if it'll help. Okay, now that means if I have more dopamine coming into the system, that's going to be inhibitory here. Right? If I'm inhibitory here, this is hyperpolarized. At this position, I'm going to have less GABA. Okay. If I have less GABA at this position, this neuron is going to depolarize, and I'm going to have an increase in GABA here. Okay. If I have an increase in GABA here, I'm going to have a decrease in glutamate here. Bless you. Okay. If I have a decrease in glutamate here, so if this is a GABAergic neuron and there's less glutamate here, what happens to this neuron? If I have a decrease in glutamate at this position, and this is a GABAergic neuron, is it going to be more hyperpolar? Is it going to release GABA or not release GABA? Not release GABA. So I'm going to have a decrease in GABA at this position. And can you see that if I have a decrease in GABA here, then this neuron is going to fire, and I'm going to increase movement. I think I might want to break that up next year. Yeah, Ben?
So the decrease in glutamate and the increase in dopamine are both contributing to the decrease in GABA.
Sorry. The increase in dopamine, so say that again, one more time.
So in the caudate nucleus.
In the caudate nucleus, yep.
So you have the decreased glutamate in the purple circle.
Yep.
Increased dopamine.
Yes.
So increase in dopamine and decrease in glutamate, are those both inhibiting the release of GABA?
Oh, I understand what you mean. So I'm thinking about, so when I say not increase or decrease, I'm talking about the relative concentration. If you have a situation where dopamine is in the lead, since dopamine is inhibitory and glutamate is less, and that's stimulatory, you're going to have an increase or a decrease in fire.
Okay.
Does that make sense?
So in our other example, when our glutamate and dopamine were both high,
That's the direct pathway.
And we had a ton of GABA, that's because it was a D1 receptor. Okay.
Yes. Yes. Okay. So, in other words, the direct pathway that's glutamate predominant, where there's not very much dopamine in the system, that's going to tend to inhibit unwanted movement. Okay? And that's all because you have so much glutamate coming into the system. Okay? An example where you have more dopamine coming into the system, where you're increasing movement, is going to be, let's say, increased movement amplitude. Okay? So that's going to be large versus small. If that makes sense. Okay. Questions? All right, now, let's look at, keeping this in mind, Huntington's is going to be the next problem, and this is going to be a problem with the indirect pathway. So we can write that up here. So Huntington's is a problem with the indirect pathway. Okay. So same old players. This is caudate. This is putamen. Okay, this is GPE, GPI, thalamus, subthalamic nucleus. Okay, and then here's your substantia nigra. Okay. You also have up above your SMA, PMC. Yeah. Okay. So here, with Parkinson's disease, there was a problem with the substantia nigra, so that you didn't have any dopamine in the system. Does anyone know what the problem is with Huntington's disease? Has anybody ever heard of Huntington's? A lot of movement. Yeah, there's going to be a whole bunch of movement. That's what the problem is with the indirect pathway. Does anybody know what causes it? What's the pathology? So what happens in Huntington's disease is you have, early on, the first structure to go is going to be your caudate. Okay? And then as disease progresses, you're also going to get destruction of the putamen. Okay? So I've drawn X's through them right now and realize what happens in the beginning. You start to get degeneration of both the caudate, and then you get degeneration of the putamen. Okay, but what I want you to think about is what's going to be the effect on this indirect pathway if I lose both pieces of the striatum. Let's kind of draw it out and see what happens. So thinking about moving, okay, you've got this neuronal cell body here, glutamatergic, and it's hooked up with your caudate nucleus, okay, and then it's also hooked up with your putamen. Okay. In there you have neuronal cell bodies that are GABAergic. Okay. These axons coming down are linked to the GPE, to a neuronal cell body in the GPE. And that neuronal cell body in the GPE is GABAergic. Coming down here makes a connection indirectly with the subthalamic nucleus, which has a glutamatergic neuron. That synapse is in the GPI with the GABAergic neuron. Hooked to that glutamatergic neuron in the thalamus, thinking about that relays the signal back to the motor planning area. And in addition, we have a situation where the substantia nigra puts out these dopaminergic neurons that are going to bind here and here. Okay. And then you have these neurons have D2 receptors. Okay. So like I said in the beginning, the very beginning of Huntington's disease, it's the caudate that is deteriorated first, and then later on in the disease process you have the putamen that's deteriorated. So we had looked previously at a situation where glutamate was predominant here. So if you have a lot of glutamate coming in this position, glutamate causes these neuronal cell bodies to fire, causing a lot of GABA at this position. Okay, a lot of GABA at this position, then this neuron's not going to fire. You're going to have this neuron released from inhibition, so you have a lot of glutamate here, right? If you have a lot of glutamate here, then this neuron is going to fire and you're going to stop unwanted movement. Okay, that's the situation where you have a lot of glutamate in the system. Now, what happens is you're losing the influence of these neurons. Okay, so not having these these neurons fire, not having a predominance of glutamate in the system is going to cause an increase of, if they have an increase of dopamine in the system. So this is going to be, the situation here is you're going to have more dopamine and you're going to have less glutamate because these structures deteriorate. Okay? If you have more dopamine, it's going to inhibit this neuron. Okay? If you inhibit a GABAergic neuron, that means you're going to have less GABA at this point. If you have less GABA at this point, that's going to cause this neuron to fire. You're going to have more GABA at this point. If you have more GABA here, you're going to hyperpolarize here, so you're going to have less glutamate and glutamate here. If you have less glutamate here, that's going to hyperpolarize this. You're going to have less GABA here. If you have less GABA here, you're going to have an increase in unwanted movement. Yes?
How's anything coming from the putamen and caudate at all if we were destroyed when that's nothing's coming from there?
Yeah. So what happens is, in the beginning with Huntington's disease, this is the first to be destroyed. So if you want to just think about half of the amount of signal coming in and it's dopamine predominant, that's another way to think about it. I'll just leave it at that. Okay. So this gets destroyed first, and then this gets destroyed over time. As the entire striatum, I can tell you with Huntington's disease, as the entire striatum gets destroyed, this patient population starts to look more like Parkinson's patients, but that's not important for this. Yes?
I guess I just don't really understand how is not active.
The caudate nucleus is destroyed. It's just going to degenerate over time. It starts to shrink. It starts to atrophy. We're going to look at that first thing.
So how would that change versus all this having more result? Like, you have that
You have what on both and get the same type of reaction.
like if there was more from the
There's more dopamine, so you're losing the influence of the, you're losing the input. If you want to start with the caudate, say the caudate is destroyed, you're losing the influence of having a lot of GABA in this area. So glutamate is usually predominant. Okay, dopamine is the modulator. You can add or subtract the dopamine. Okay. But what you're losing in this situation is Huntington's disease in the very beginning is when you lose the caudate, you're losing the influence of the glutamate in the system.
okay, so there's
There's
just less.
less glutamate. Yes. Less glutamate. Less glutamate. Well, you have the glutamate being released, but the glutamate can't work because that structure is being destroyed. And you'll see that in a minute. Okay, other questions? It's really a matter of thinking about the amount of neurotransmitter at each of the positions. And I think if you walk your way through, it'll start to make sense. So what I'd like to do now is let's go ahead and